So starting on chapter 3, the first person it talks about, and remember when we go through the history on any of this, you're not going to be asked to talk about years or anything like that. I just want you to understand how each person contributed to the growing body of knowledge. So now we are jumping back to before the experiments that we talked about in the chapters on DNA structure. And this is back before they knew what DNA was and how they were trying to figure out how inheritance worked at all. So there was a guy named Gregor Mendel that was a monk who worked at a monastery and studied pea plants. So it might seem weird to us that he would be studying pea plants when he's working at a religious institution, but it happened that at that time, most of the most well-educated people worked at monasteries because they were in an area where a lot of people couldn't read. So to be able to have that kind of skill already puts you in a different class. It happens that he studied pea plants over the course of 7 years and about 28,000 plants. Peas are great because they could work with them in a small plot, and then they could eat them afterwards. So very, very helpful to if you're going to be researching something. So he's got his pea plants going, and but this kind of came about because prior to this kind of discovery, what they believed was going on is there was a blending hypothesis, and that said that from one generation to the next, the two traits are going to be blended together. If you have a tall person and a short person, they get married and have kids, they're going to have medium-sized children. But if this were the case, there wouldn't be much variety in the world at this point because everything would have all been bred out to be just blended. And we do sometimes see this. Whenever you have codominance and incomplete dominance, you end up with these in-between blended states. Like if you had a white flower and a red flower and you end up with a pink flower on the next generation. That's a type of incomplete dominance. But what Mendel was seeing wasn't that sort of incomplete dominance. What he did was he actually went against these ideas and used mathematics. And remember that there's quantitative and qualitative. Quantitative is a quantity of something. So if you're counting numbers, it's a quantitative answer. Qualitative is looking at like the color of something. So if you were comparing two test tubes and you say tube B is darker than tube A, that's a qualitative answer. If you put it to the spectrophotometer and measure the absorbance, then you get a quantitative answer. So he was using quantitative analysis to actually go through and study this very, very systematically. And when he first presented this information, I mean, he's known as the father of genetics, but it was completely ignored until it was rediscovered in the early 1900s. Now, about the same time that Mendel was working on this, Darwin was writing his Origin of Species, and he knew like that he could look through and they could see the chain of evolution, but they didn't know what was going on to exactly pass a trait from one generation to the next, to the next, to the next. And all of the questions that he had could have been answered by Mendel, but he didn't read Mendel's paper. They actually found in Darwin's office the journal where Mendel had published his paper. It's in there, but they could see that the seal was never broken on it. So it was very like so close, and they could have put it all together back then, but they never got in touch with each other. And it happened that after Mendel published his paper, which his goal wasn't even to really look at genetics, he was looking at plant hybridization to try to help people who were trying to grow plants. He didn't know anything about the genetic side of it. It was just okay, well, you breed this one with this one, this is what comes out, and that could help farmers. But then he tried it again with some other experiments and types of plants, but it turns out that they were ones that were not self-fertilizing. They didn't do sexual reproduction, so they didn't have dominant recessive traits to pop out the same way that the pea plants did. And so that didn't work out so well. It left me with even more questions. And in the meantime, he was promoted. So he was now running the Abbey, and it became just that he didn't do any more research. So he wasn't involved in any of the further discussions about genetics that were going on at this time period. So then his work was rediscovered by 3 or 4 different people all in the same year, right at the turn of the century. And they were like, oh crap. They understood the significance of what he had done. But here's Bindle. So just giving you a little background on how it works. Remind y'all that gametes are the haploid reproductive cells. You have the sperm, which are basically the pollen grains that are produced in the anthers. And then the female gametes, they're the eggs, are within the ovules of the ovaries. So you usually have to have the pollen land on the stigma, migrate down through to fuse with the egg cell. So what he would do actually is he would go through and remove the anthers from the flower so that it couldn't self-fertilize if he didn't want it to. And then he would use a paintbrush to transfer the pollen onto it from another plant. And it would land on the sticky part here up the top of the stigma, travel down, and form the egg, or fuse up, and then an egg forms. So you don't need to know any of the anatomy on it. I just want you to see how he was able to control between self-fertilization, where the pollen and egg are derived from the same plant, and cross-fertilization, for where they're coming from one plant to another. So if they were going to be self-fertilizing, he could actually cap them off so that the flowers weren't exposed to any other pollen. And that's how he kept everything controlled, because they were outside and pollen just blows in the air. So he kept them covered. All right. So he was studying 7 different characteristics for height, flower color, flower position, seed color, seed shape, pod color, and pod shape. So most of these are pretty self-explanatory. You could look. And he started off studying them 1 at a time, and then 2 at a time. So to begin, he made true breeding lines, which are plants that always produce progeny with the same traits when they're self-fertilized. So if you had a tall plant, every single generation, no matter how far you went back, all tall plants, tall plant, tall plant, tall plant, dwarf plant, dwarf plant, dwarf plant. Well, that's tongue twister. Or all yellow seeds, always yellow seeds, that kind of thing. So once he was sure that they were going to be true breeding, then he could start crossing them with each other. So they have some notes here for the terminology that a character is basically a type of characteristic that can vary. So like height. So he was studying 7 different characters. Then you had traits or variants, which were versions of the character. So like if you were studying the trait or the character of height, you would have a trait or variants that were tall and dwarf. We now know that we usually call these alleles based on the gene that is shown. That goes with DNA sequencing that they didn't know about back then. So he goes through, starting off with his single factor crosses, and starts using his empirical approach to follow through very systematically. And he starts creating hybrids. So if you're looking at 1 character at a time, it's called a single factor cross. So if you're looking at just plant height, if you're looking at 2 characters, it's called the 2 factor cross. So that would be like seed height and plant or pod color, something like that. So first ones, what he would do is have the P generation, the parental generation, in this case, tall and dwarf. And so if we look at the genotype, which he didn't exactly have this drawn out, he didn't know what was going on yet, big T, and little t, little t. So anything that is true breeding, it needs to be homozygous dominant or homozygous recessive. It can't be heterozygous. So we cross our big T with our little t's, and we get mismatched ones, big T, little t. So all the hybrids. So those become the F1 generation, the first filial generation. Those seeds get planted, they grow up, and he looks at them and takes their measurements. Then he allows those to self-fertilize. So the heterozygotes, big T, little t, are crossing. And he was getting 3 tall plants and 1 short plant. we now know that if we look at their genotypes, it was 1 big T, 2 heterozygous big T, and 1 little t. But that was how it worked. P generation to F1 to F2. And so looking at his data, he found that every time he crossed 2 different traits, only one showed up in the F1 generation. So if we cross tall and dwarf, on the F1 generation, they are all tall. But then when you got to the F2 generation, that other trait would pop back up. And it was always at a 3-to-1 ratio. So if you look at 2.98 to 1, pretty darn close to 3, though. So he was like, all right, something's going on here. So what I always thought was interesting is that he discovered 7 traits that were completely independent of each other with no sort of incomplete dominance or codominance or linkage, or like the statistical odds that he did that were just crazy, considering pea plants have 7 chromosomes and he was studying 7 traits. So you would think that there must have been 1 gene on each chromosome, and that's why there was no kind of linkage, crossing over type effects that we haven't learned about yet. But it turns out that these were all on, so like of them, they were on 4 of the chromosomes, but they were spaced so far apart that they all acted as if they were independent of each other. So we'll go in and we'll review meiosis and find out what was going on there. But here we can see the other thing I thought was, well, maybe he was actually like just throwing out, like maybe he looked at like 20 traits and these are the good ones, but they actually have his original journals, and he just happened to be very lucky when he picked his traits so that he didn't have any data that was conflicting. So again, just repeating, of the 7 characters studied, of the F1 generation, only one of the 2 parental traits was there. On the F2, they would end up with a 3-to-1 ratio. So he was going, okay, this one of the traits is disappearing during the F1 generation, but when it reappears, it's completely unchanged. So before, when the idea was blending theory of inheritance, so if you had like that red and white flower cross and they'd end up with pink, it wouldn't go to the next generation and then have white or red suddenly pop up totally like that. It was so he goes, okay, what is going on here? So they had the idea that there was some sort of particle that was going on, a particle that was passing from one generation to the next. We now know these were chromosomes and genes, but back then he didn't know that. So he started using the terminology that the trait that was exhibited in F1 was the dominant, and then the trait that was masked in F1 was called the recessive. So that 3-to-1 ratio was always 3 dominants and 1 recessive. So he went through and he put this together to say, all right, the genes are segregating from each other during a process that gives rise to the gametes. And he called this the law of segregation, that they were separating out in some way, and that they were not blending, they were not mixing together. So the 2 copies of a gene segregate or separate from each other during transmission from parent to offspring. That's the whole basis there for law of segregation. This is a reminder for any terminology being used here. So genes are the unit of heredity that we now know as DNA sequences. The alleles are different versions of the same gene. Homozygous is when there's 2 identical alleles. Heterozygous are 2 different alleles. A genotype is an individual's specific allele composition. So if you're looking at the DNA, which DNA is there? The phenotype is the outward appearance of the individual. So if something is heterozygous or homozygous dominant, you can't tell based on their phenotype which one they are. They look the same, since all you see is dominant trait. So again, we have our P generation, that is our tall plant. We get our segregation, but because the tall plant can only pass on big T, we know we're passing on a big T. Our dwarf plant can only pass on little t. So it's going to give one big T, one little t. Then on the self-fertilization, now all of this is going to get remixed around. And you sometimes had that big T crossing with the big T to give us our double dominant plant. Sometimes you had your big T crossing with the little t here. That gave us our heterozygous tall plant. Now we'd have this little t crossing with this big T to get our heterozygous tall plant. And sometimes you had the little t and the little t coming together to give us our dwarf plant. So Reginald Punnett was the geneticist who actually made the little squares that we now know as Punnett squares. So using this information here, if we wanted to make a picture of this, we need to write down the genotypes of both parents, write down the possible gametes that each parent can make. So all right, so gonna make us a grid. So up here on the side, we have the dad side, big T, little t. And over here we have the mom side. She's also heterozygote, but these are her options. So now if we drop these here, big T, little t, little t. And now coming across, big T, little t, little t. And I like drawing it this way to get the big than little, but it works also even if you have your little t and your big T here. It doesn't matter because it's just heterozygote. So we can tell now that, going back over to PowerPoint, this is what they draw out, much prettier than mine. And we can see now that our genotypic ratio is 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. But our phenotypic ratio is 3 talls and 1 dwarf, because anybody who has a big T, that is going to show up because it is dominant. In order to get a recessive to show, you have to have 2 recessives and no dominant present. So then he started going, all right, that's fine and dandy. Let's look at 2 factors. So in this case, you were crossing seed shape, round and wrinkled, and seed color, yellow versus green. And that gives, there were 2 possible patterns. They could be either linked together, that they couldn't be separated from each other, or they were independently assorting. And so what, on one side, they have round and yellow with wrinkled and green. And when they were crossed together, we get our F1 generation that is heterozygous. And if they are linked together, you don't get any reassortment. It has to be only yellow or only round and yellow and only green and wrinkled. But it can't have a mix that's going to be like round and green. Or the other hypothesis is that after they combine together, you could end up with all 4 characteristics being possible. And that would be if they were independently assorting. And so he did the same thing. He did his true breeding round yellow seeds, combining with his true breeding wrinkled green seeds. They get planted, so they get their appropriate plants. We cross the pollen from one to the egg of the other. We get our F1 seeds. We allow those to self-fertilize. And now he found a weird thing happening. he was always getting 9331, meaning that there were 9 round yellow ones, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. Always got those odds. So you can see here the ratios. And so now what we were finding is the F2 generation was containing novel combinations not found in the parental generation, the round in green and the wrinkled in yellow. So these non-parentals are predicted if the genes were segregating independently of each other. So this now comes up with the number 9331. And I'm not going to redraw out that Punnett square, but you can see here what they did. You have your choice. Are you going to pass on a big R or little r? So half the time you're passing on big R's, half the time you're passing on little r's. Are you going to pass on your big Y or your little y? Y. Half the time you're going to pass on big Y, half the time you're going to pass on little y. And ditto for dad. And then you can just draw everything out from there. You're going to play with this in your homework some, I believe. So I'm not drawing it out. But so his law of independent assortment says that 2 different genes randomly assort their alleles during the formation of haploid cells. Now, this is only going to work if you're looking at genes that are not linked together. He didn't know anything about linkage, so he didn't know to add that kind of caveat to the rules. But again, we can do the Punnett squares, but we would have to have 16 boxes. And we could draw it out in this way to see, again, 9331. If you were to go to 3-factor crosses, you could do it, but you would need 64 squares in your Punnett square, so you wouldn't really want to do that. So usually they did a multiplication method or a forked line method to show that. So I'm not going to ever ask you how to do 3-factor crosses, so we don't have to worry about it too much. But if you was always, you're always looking at 3/4 and 1/4, this is what we get. At the end of the chapter, it talks about statistics, and so we're going to see where they're getting these numbers from then. So I'm going to pause video so it'll split